Welcome, medical peers and students, to this advanced explainer. Today, we're setting our sights on a truly fascinating deep dive into the absolute elegance of molecular endocrinology. As clinicians and researchers, we know that understanding how our cells communicate isn't just, basic science, it's the absolute foundation of everything we do at the bedside. So bring your academic focus because we're going to trace the incredible journey of hormones right from their initial molecular binding all the way to their widespread pathophysiological effects. Okay, let's dive into this. Hormones are essentially the master conductors of our body's symphony. They don't just gently suggest that a cell should do something. They act as critical messengers that strictly orchestrate everything from our baseline metabolism to complex reproduction and growth. Mastering these cellular conversations is absolutely critical for our clinical practice. Why? Because when this orchestration falls out of tune, that's exactly when we see systemic disease. So here is our clinical syllabus for today. First, we'll cover molecular mechanisms of transduction. Second, we'll contrast membrane and nuclear receptors. Third, master signaling feedback loops. Fourth, we'll use glucose regulation as our clinical model. And finally, we'll wrap up with the pathophysiology of endocrine dysregulation. All right, starting right off with Section 1, Molecular Mechanisms of Transduction. The fundamental concept to grasp here is that a hormone's exact molecular structure directly dictates its function. It dictates exactly which specific target cell receptors it can even bind to. Once it reaches that target, it triggers a rapid, sequential cascade. Think of it like a cellular domino effect. Step 1 is receptor activation. This physical binding transduces the signal, leading to Step 2, the generation of second messengers, like cyclic AMP or IP3. Step 3 involves these messengers triggering incredibly complex intracellular signaling cascades, which often involve protein kinases. And finally, Step 4, the ultimate cellular response. This could be altering cellular metabolism right then and there, or upregulating gene transcription. So the receptor is essentially translating a single extracellular message into this massive intracellular regulatory network. Moving on to Section 2, membrane versus nuclear receptors.
Now, what's really interesting about this slide is the stark contrast in both timing and mechanism. On one side, we have membrane receptors, like G-protein-coupled receptors. These live right on the cell surface and trigger those second messenger cascades we just talked about in a matter of milliseconds. Their effects are incredibly fast. Actually, scratch that, they are almost instantaneous. On the flip side, we have nuclear receptors, which are ligand-activated transcription factors. Hormones like the thyroid hormone actually have to enter the cell, bind to these receptors, and then translocate to the nucleus to interact with specific DNA sequences. This DNA binding mechanism is much slower, sometimes taking hours or literally days to manifest, but it provides highly prolonged and sustained gene regulation.
Let's zoom in on a very specific and, frankly, crucial type of membrane receptor, receptor tyrosine kinases, or RTKs. Upon ligand binding, these receptors undergo this fascinating process called autophosphorylation. This specific activation triggers massive downstream cellular events that govern proliferation, differentiation, and survival. RTKs play a huge role in growth factor signaling. I definitely want you to keep this mechanism in mind because it sets up the exact targets we look for in advanced cancer therapies a bit later on.
Next up, Section 3, master signaling feedback loops.
So, stepping back from individual cellular receptors and looking at systemic integration, let's walk step by step through the hypothalamic pituitary adrenal, or HPI, axis. This is the prime example of a finely tuned negative feedback loop responding to autonomic nervous system stress. First, a stressor causes the hypothalamus to release CRH. Second, this triggers the anterior pituitary to release ACTH into the bloodstream. Third, ACTH travels down to the adrenal cortex, stimulating the release of cortisol. And here is the absolutely crucial fourth step. That circulating cortisol then travels right back up to negatively inhibit the further release of CRH and ACTH. It effectively shuts off the stress response and returns the entire body to baseline. But what happens when that feedback loop actually breaks? If a patient experiences chronic, unrelenting stress, or say they're on long-term glucocorticoid medications, it causes severe HPA axis dysfunction. The body just becomes flooded with cortisol. It's like a biological fire alarm that literally won't shut off. This leads to Cushing's syndrome, which is characterized by massive downstream systemic effects, including severe cardiovascular implications like extremely high blood pressure, as well as metabolic disasters like type 2 diabetes. Which brings us to Section 4, our clinical model of glucose regulation. To really understand this pathway integration, we're going to use the insulin and glucagon cycle as our model. I want to pose this question to all of you. How do these intricate signaling pathways actually crosstalk to maintain physiological equilibrium? In the dynamic landscape of cellular signaling, pathways rarely ever act in isolation. For instance, think about how the AMP-activated protein kinase pathway and insulin signaling intricately crosstalk to govern energy balance and glucose homeostasis. It is a continuous, highly dynamic negotiation within the cell. And the results of that cellular negotiation must be incredibly precise. Just look at these numbers. The endocrine system has to maintain blood glucose in a very narrow window of homeostasis. We're talking ideally between 80 and 130 milligrams per deciliter right before a meal and under 180 2 hours after eating. If the pathways fail to integrate properly, the patient rapidly slips into either tissue-damaging hyperglycemia or immediately life-threatening hypoglycemia. So the crucial point is this constant push and pull molecular mechanics between the pancreatic islet cells. When glucose is high, beta cells secrete insulin, which causes GLUT4 receptors to translocate to the cell membrane to pull glucose inside for glycolysis. It also directly inhibits lipolysis and ketogenesis. Conversely, when glucose drops, alpha cells kick in and secrete glucagon, which stimulates the liver to create new glucose via gluconeogenesis and break down stored glycogen. Glucagon is basically our biological failsafe, preventing fatal hypoglycemic drops. This push-and-pull molecular mechanic also dramatically builds out our diagnostic capabilities. When beta cells produce insulin, they cleave it from a much larger pro-insulin molecule, which leaves behind a byproduct called C-peptide. Because insulin and C-peptide are secreted in exact equal molar amounts, and because C-peptide has a much longer half-life with far fewer fluctuations, we use it clinically all the time. Measuring C-peptide allows us to reliably differentiate endogenous hyperinsulinemia, which we often see in early-stage type 2 diabetes, from exogenous insulin. It gives us a beautifully clear window right into their residual beta cell function. All right, final section number 5, pathophysiology of endocrine dysregulation. We've looked at healthy homeostasis, but now we have to examine the bedside reality of what happens when these pathways fail. Signaling dysregulation results in cascading, widespread maladies. We see the direct overproduction or underproduction of hormones, leading to conditions like Addison's disease or acromegaly. Aberrant receptor activation can cause uncontrolled cell growth and cancer. Disruptions in crosstalk lead directly to metabolic syndrome and insulin resistance. And sometimes, the immune system just attacks the pathways themselves, like in Hashimoto's disease, where antibodies actively target thyroid peroxidase or thyroglobulin, practically shutting down the entire metabolic chain. And this brilliantly illustrates exactly where the future of endocrinology is heading. Modern genomics and molecular testing are fundamentally changing how we approach these exact dysregulations. By uncovering the specific genetic basis of endocrine diseases, we can now detect genetic carriers so much earlier. We can precisely diagnose the exact molecular subtype of a disease, rather than just throwing treatments at the broad physiological symptoms. This brings us right back full circle to the concept of targeted therapies. Remember those receptor tyrosine kinases we discussed at the very beginning? By understanding the precise molecular events governing their activation, we've developed tyrosine kinase inhibitors. These drugs specifically disrupt those aberrant signaling cascades in conditions like cancer. We have officially closed the translational loop, from understanding the molecule directly to treating the malady. So, I'm going to leave you with this final provocative thought. As genomics continue to map out the exact molecular crosstalk of our hormones, how will these targeted therapies completely reshape your differential diagnosis on the ward? The era of broad systemic treatments is rapidly giving way to highly precise molecular interventions. I strongly encourage all of you to keep applying these microscopic insights to your macroscopic patient care. Keep questioning, keep studying, and keep connecting the molecules to the medicine. Thank you so much for joining me for this explainer.